the WI38 cell line: Biological Concepts, Medical Impact, and Societal Relevance, by Kellen Smith for MCB 150. First, an introduction to the WI38 cell line. The WI38 cell line is a line of human cells originally derived from lung tissue in the early 1960s. It became one of the most important tools in medical research. The purpose of this presentation is to explore three main aspects of WI38: the biological basis of the cell line, its medical impact, and its societal and ethical significance. And I hope to answer the question, how did this one scientific discovery transform global health and raise lasting ethical questions? So first, starting with cell division. Cell division, or mitosis, is the process by which a single cell duplicates its DNA, gives and divides to form two identical daughter cells. It includes stages such as prophase, prometaphase, metaphase, anaphase, and telophase, followed by cytokinesis. This process is critical for growth, repair, and maintaining healthy tissues. In the context of cell lines like WI38, understanding mitosis is essential because these cells can be cultured to divide many times under controlled conditions, which allows scientists to study human biology without needing to repeatedly collect new tissue samples. So what is mitosis? Mitosis is the process of creating two new daughter cells, and it works by first replicating DNA and then condensing that DNA into chromosomes. Then the nucleus breaks down, and microtubules move the chromosomes to the center or the metaphase plate, then pull the sister chromatids apart, reform each nucleus, and then cytokinesis divides the cytoplasm into two new daughter cells. The origin and characteristics of the WI38 cell line. The WI38 cell line was developed in 1962 by Leonard Hayflick and Paul Moorhead at Wistar Institute. It was derived from the lung tissue of a legally aborted female fetus of about 3 months gestation. WI38 is composed of normal human diploid fibroblast cells, meaning the cells have a normal set of 46 chromosomes. Unlike cancer cells, WI38 cells have a finite lifespan. They can divide only about 50 times, which is a concept known as the Hayflick limit. These cells are carefully maintained in lab conditions, meaning they were kept at body temperature, given nutrients, and grown in sterile culture flasks. Because they are genetically stable and free of animal contamination, WI38 became a gold standard for producing safe vaccines. Next, it contributed largely to vaccines. So WI38 made an enormous impact on medicine by providing a clean, human-based system for growing viruses used in vaccines. Before WI38, many vaccines were produced using animal cells, which sometimes introduced unwanted contaminants. WI38's human origin made it safer and more effective for human vaccines. One of the most notable successes was the rubella vaccine. The virus that causes rubella, or German measles, was grown in WI38 cells to develop a vaccine that has prevented millions of birth defects worldwide. Also, on the side I have a chart, and you can see that around 1960, people stopped using antibiotics and the rate of using vaccines starts to skyrocket. And then next, other vaccines besides rubella, some more specific examples. So beyond rubella, WI38 was also used in developing vaccines for several other diseases, including rabies, measles, mumps, varicella, chickenpox, adenovirus, and polio. So first, for rabies, WI38 was used to develop human diploid cell rabies vaccine that replaced older, unsafe versions made from animal nerve tissue. The human-based cell vaccine caused fewer side effects and provided longer-lasting immunity. It became the global standard for rabies prevention recommended by the World Health Organization. Next, measles and mumps. Measles and mumps for WI38 cells were used to propagate and test strains of measles and mumps viruses, ensuring vaccine safety and consistency. These vaccines became part of the combined MMR vaccine, which is for measles, mumps, and rubella. This continues to protect billions of people worldwide. And then finally, chickenpox. The varicella zoster virus, which causes chickenpox, was grown in WI38 cells to produce the first safe and effective vaccine against this disease. Licensed in the 1990s, this vaccine drastically reduced hospitalization and death rates among children. So WI38 isn't just a research tool. It's a direct reason why many of us are vaccinated and protected today. The vaccines are used in over 100 countries and have been critical in the near eradication of diseases once considered inevitable. So while the biological foundation of WI38 was crucial, its true power lies in how it transformed medicine and public health. But behind this success, there's also a complex and sometimes controversial human story, which brings us to the history and ethics surrounding WI38. First, we'll discuss the history. So the WI38 cell line was developed in 1962 at the Wistar Institute in Philadelphia by microbiologist Leonard Hayflick and his colleague Paul Moorhead. They isolated the cells from the lung tissue of a legally aborted female fetus that was approximately at 3 months gestation. At the time, scientists were searching for reliable and uncontaminated human cells to grow viruses for vaccines. Because, as I said earlier, existing animal cell lines sometimes carried hidden viruses that could cause disease. The discovery of WI38 provided a breakthrough because these cells were human diploid fibroblasts. Also, as I said earlier, this means they contain the full set of 46 chromosomes. Also means they were genetically stable, unlike cancerous or immortalized cells people used before. Hayflick and Moorhead published a groundbreaking paper in 1963, introducing the concept of the Hayflick limit. This was the discovery that normal human cells can only divide about 15 times before they stop reproducing, which contradicted the long-held belief that all cultured cells could divide indefinitely. This finding reshaped the scientific understanding of cellular aging and mortality, making WI38 not only a practical tool for vaccine development, but also a model for studying the biology of aging. In the years that followed, WI38 became one of the most widely distributed and studied human cell lines in biomedical history. It used to produce the first safe and effective rubella vaccine in the 1960s and later contributed to vaccines like we talked about earlier. Despite its success, the use of fetal tissue to create WI38 raised ethical debates that persist even today. It sparked conversations about consent, ownership, and the moral dimensions of scientific progress. But, nevertheless, WI38 remains one of the most significant and influential cell lines in medical history. Moving on to the ethics. As I just said, WI38 brought medical breakthroughs, but it also sparked ethical debates that are still very present today. First, the cells came from a fetus aborted in Sweden in 1962, and at the time there was no informed consent process for tissue use. So it's not known if the mother of the fetus gave consent or even if she was aware that her fetus was used. This raised difficult questions like who owns biological materials, should consent be required for research use? And for many, the use of fetal tissues pose moral challenges, especially in connection with religious or personal beliefs about abortion. These ethical discussions have influenced policies on biomedical research and continue to shape how new cell lines are developed today. And next, the public health impact. From a public health perspective, WI38 revolutionized how vaccines are made and tested. Its success demonstrated the value of human cell lines for disease prevention. It also led to stricter guidelines on how human-derived materials are collected and used, helping to ensure greater ethical transparency and safety in modern research. In short, WI38 not only saved lives but also reshaped public health policy and the ethics of scientific practice. And a personal reflection on the impact and the ethical stance. Personally, I think WI38 represents both the accomplishments and complexity of medical science. It shows how one discovery can transform global health outcomes, but also how progress can raise difficult ethical questions. On a personal level, it's incredible to realize that vaccines I've received were likely developed with the help of this cell line. And ethically, I believe human-derived cell lines can be used if handled with respect, transparency, and consent. Research should always balance scientific benefit with moral responsibility. The ethics of using the fetus is a difficult line, but in the end, it helped over 10 million people and saved countless lives in the process. In conclusion, the WI38 cell line is biologically important because it helps scientists understand cell aging and provided a safe system for studying human cells. Medically, it was a foundation for some of the most successful vaccines ever developed. And societally, it has shaped our ethical frameworks and public health policies. WI38 is a reminder that science, ethics, and humanity are always connected, and that discoveries made decades ago still protect and influence us today. And here's a list of references.